So quick little bio. I'm Adam. I'm a postdoc in Adeen Flinker's lab at the NYU Med School. Did my PhD with Vic Ferreira at UC San Diego and master's with Matt Wagers in linguistics at UC Santa Cruz. But the main focus of my research nowadays is on the neuroscience of sentence production. So how the brain translates from thought to language. And even though in many ways this is where the field started, so I'm showing you the brain that made Paul Broca famous, compared to research on comprehension, there's actually been very little work on production. And I think there are two main reasons for this. The first is just that it's really difficult to get people to say what you want them to say. So for instance, if I ask you to describe the scene, you might say what I want you to say, the nurse is spraying the chicken. But as pretty much anyone who's run a sentence production study will tell you, even for the simplest sentences, people will come up with creative ways to say pretty much anything but what you want them to say, surprising you with passives when you want actives, saying duck instead of chicken, going into way too little detail or way too much. And a consequence of this is that the production research is largely focused on single words. And these are just much easier to reliably elicit. So this typically involves picture naming paradigms where participants see a picture of, say, a chicken and say chicken. And between a lot of theoretical, behavioral, and neuroscientific work, we've converged on some facts about words, which probably everyone here knows, but I'll run through them. So first and foremost, words are not unitary objects, but conglomerates of representations, including meaning, grammatical properties like gender, phonological information, as well as articulatory and perceptual representations. And each of these representations is associated with a particular part of the brain, and the specifics here don't matter. This is just to give you a sense of the level of granularity we've arrived at in studying words. And to highlight that we really don't have anything like this for sentence production. So we do have really good theoretical models, and again, specifics here don't matter. This is just to demonstrate that we have a good sense of sort of the computational level of what's going on during production. And we even have neural models of particular parts of this. So as I already showed you, here's sort of where we are with words, and there's also models of syntax in the brain. But much of the rest of this remains unexplored, including, for example, how syntactic positions like subject and object are encoded. Okay, so we're far from a holistic neural model, and the question that I want to address today is not exactly what does this look like in the brain or what are the neural dynamics of sentence production, but something even more basic, which is can we leverage what we already know about words as a starting point for understanding sentences? So specifically, are the neural dynamics of word production during picture naming the same as the dynamics of word production during sentence production? Now, I mentioned that there were two hurdles that have impeded progress in the neuroscience of sentence production. One was the difficulty of controlling what participants say, and the other is technical. So even with the advent of non-invasive methods, studying language production was still very difficult due to the sensitivity of these methods to motor artifacts. And a major advance has been the use of electrocorticography, or ECOG, to study language production. So ECOG is the recording of electrical activity directly from electrodes implanted on the cortex in neurosurgical patients. I'm about to throw up some neurosurgical pictures, so if anyone's squeamish, maybe look away for a moment. But in practice, what this looks like is we have patients, typically with epilepsy, who spend sometimes up to a week in the hospital with their skulls off and grids of electrodes like this implanted on the surface of their brains, continuously recording electrical activity. These recordings are virtually immune to motor artifacts, and unlike traditional electrophysiology and neuroimaging, ECOG provides high spatial and high temporal resolution. That is, you know exactly where the electrodes are and exactly when a spike in electrical activity occurs. So in this study, we collected ECOG from 10 patients with electrode coverage in traditional language areas in the left hemisphere while they performed a language production task, and these are just all the electrodes from all the patients collapsed onto one brain. The task had 3 blocks. First, they completed a picture naming task where they'd see a picture and name it. So here they'd say chicken, and here they'd say Frankenstein, and so on. And the whole experiment involved only 6 characters, chicken, Dracula, ninja, dog, Frankenstein, and nurse. And they had been familiarized with these characters before the experiment in a quick training block. I should also mention that this experiment was not designed for the purposes that I'm going to talk about today. It was originally planned for something else that failed pretty miserably. But we got some cool data, and what I'm showing you are results from some exploratory analyses. Okay, so after the picture naming block, participants performed a sentence production block, and here they would see a question that they were instructed to read aloud, who is spraying whom, and 1 second later they saw a scene involving 2 of the 6 characters, and they'd say a sentence like the nurse is spraying the chicken. Okay, so let's start by looking at some word production data. Here I'm going to plot neural activity over time, and throughout the talk I'm using activity in the high gamma band as a proxy for neural activity. This correlates highly with neuronal firing and bold signal. And also in the plots I'll show you I'm time locking to speech onset. So time 0 is when the patient begins to speak. And in this plot, I'll show you the average neural activity from one electrode for each of the 6 nouns. So for example here, the green line shows the mean activity for Frankenstein, the red line for all the ninja trials, et cetera. And just by glancing at this, you can tell that this electrode shows word-specific patterns of activity. It seems to be especially tuned to the blue line, Dracula. So what we're going to do is leverage differences like these to predict what word a patient is saying on each trial. So we used a classification approach with tenfold cross-validation, and I'll break that down for anyone who's not familiar. So we started by taking 90% of the picture naming trials. Here I'm just showing you simulated data, so these are made up. And the rows here are grouped by trials, or rows, and they're grouped by which word the patient is saying here at time 0. And we pick a 50 millisecond time window here, just negative 650 to negative 600 milliseconds, the black bar, and use the data in that window to train a classifier. And a classifier is just a model that learns the unique patterns of activity for each of the 6 words. So for those of you who are more familiar with these analyses, we did a one versus rest logistic regression here. And for everyone else, doesn't matter. The intuition is just that this is a fancy way of identifying patterns of activity across the brain for each word in the black 50 millisecond window. And then we take the remaining 10% of picture naming trials, so this is unseen data, and use the classifier to predict which word a patient is saying by determining which of the 6 word-specific activity patterns that we identified the test data are most similar to. And we do this for every trial and every time sample, which at every trial and time sample gives us a prediction, which we score 0 if it's incorrect and 1 if it's correct. And then finally, by averaging all these 0s and 1s across trials, we get the mean prediction accuracy over time. And so this bottom plot is really important to understand because you're going to see a lot of plots like this in the next few slides. So the y-axis is the mean accuracy, or proportion of trials that we correctly predicted, where we correctly predicted what word the patient was saying. And the vertical black bar again shows the training window, the time the training data came from. And the gray line and shaded distribution show the mean and 95% confidence interval of the chance distribution. So this is the accuracy you get when you perform this whole pipeline 1,000 times on randomly shuffled data. And we do this whole thing for each of the 50 millisecond time windows spanning from negative 750 milliseconds, so that's before speech onset, to 250 milliseconds after speech onset, for each of 7 regions of interest and for each of 10 patients, which gives us a total of 1,400 classifiers, which means we are almost guaranteed to find something significant, so we're in good shape. I'll show you a couple of those significant cases. So here are the results from a sample classifier trained on data from electrodes in one patient's posterior temporal lobe. This is just like the bottom plot in our analysis pipeline. It shows the mean prediction accuracy across trials. And even though the classifier was trained on data from negative 350 to negative 300 milliseconds, which is the white vertical bar, already by about negative 500 milliseconds, we're able to decode what word they're going to say above chance. So that's denoted by the pink highlight. And similarly, we're able to decode for a few hundred milliseconds after the training window as well. Okay, here's another sample classifier. This one was trained on electrodes in sensory motor cortex in a different patient. And these are just 2 examples of the 444 significantly predicted classifiers that significantly predicted word identity. And rather than showing all of these to you individually, I'm going to squish them and stack them to show you the aggregate pattern. So over here on the left, I'll do this for the prediction accuracy from all of these 444 classifiers that had significant results. And as in the plots on the right, the pink highlights for each line show where the prediction accuracy was above chance. These prediction accuracies are sorted vertically by their training window time, the white bars, so later training windows are on top and earlier ones are at the bottom. And at the very bottom is a density plot showing the number of significant detections over time. Okay, so the main point here is that we are able to decode what word a patient is going to say, and in most instances long before they say it. So this whole analysis was more or less a proof of concept. It was a sanity check. It shows that we can successfully predict what word a patient's saying during picture naming using data from picture naming. Okay, now for the interesting part. We want to know whether whatever information these classifiers are picking up on and using to decode, is that information also involved in producing these words in sentences? And to address this, we used largely the same procedure, but with a couple of tweaks. So rather than training the classifiers on 90% of the picture naming trials, now we're going to train the classifiers on all the picture naming data. And then rather than testing on a subset of the picture naming data, we use these newly trained classifiers to decode words during the production of sentences, like Dracula's hitting Frankenstein. And here I'll show you the results, time locked to the onset of each of these words. Now, the sentence on top is just a sample sentence. In reality, we performed this analysis for all the sentence trials, and what I'm going to show you is the mean prediction accuracy across those trials. And here I'll show you the results from electrodes in sensory motor cortex, averaged across patients. Each one of these plots will have two lines, a blue one representing the accuracy of predicting whatever the subject of the sentence was, Dracula in the example, and a green line for the object, Frankenstein in the example. And I'm highlighting where prediction accuracy was significantly above chance. So we can see that in sensory motor cortex, we detect the subject, Dracula, right around the time that the training data came from in picture naming. And if we now look at predictions during the next word of the sentence, we see no significant prediction of either word. So whatever representation is captured by the models at 50 to 100 milliseconds in sensory motor cortex, it's just not online here. Next, for the verb, hitting in the example sentence, we see a different pattern. Around 300 to 400 milliseconds after the onset of the word, we start to detect the upcoming word, Frankenstein. And this is probably not decoding of the word in advance, as patients are most likely done saying the verb hitting in about 300 milliseconds. So the significant portion of this plot probably reflects decoding of Frankenstein while it's being said. And consistent with this, we see significant prediction of the object, Frankenstein, during its articulation. So this sample region, sensory motor cortex, shows what we're calling temporally congruent decoding. That is, it decodes the subject while the subject is being said and the object while the object is being said. But remember, this is just data from one region, and to look at the whole picture, we can again stack the results from all significant classifiers to look at the overall pattern. And here I'm just doing this for the time leading up to and following the subject noun, and as you can see in the density plot, we've got a lot of detections of the subject, the blue lines, but we're not seeing much of the object yet, the green lines. And now, if we look at the second noun, the object, we're seeing a similar pattern, very few blue lines, but lots of green lines. So, okay, so far we can say a couple of things. First, we've shown that word representations involved in picture naming do, in fact, generalize to sentences. Remember, we're decoding these words using classifiers trained only on picture naming data. And that means that the brain's way of encoding words is shared across these different types of language production tasks, which is probably not super surprising to anyone in this room. But second, the temporal properties of word information are pretty similar to those in picture naming. We find the most information right around speech onset. But now, let's look at a slightly more complex case of sentence production. So something I skipped over in the methods was that on half of trials, we prompted patients with questions with active voice syntax, who spraying whom, eliciting responses like the nurse was spraying the chicken. But on the other half, we asked questions in passive voice syntax. So who was being sprayed by whom, eliciting the chicken was being sprayed by the nurse, most of the time anyway. The analyses I just showed you only looked at active sentences, and this is the canonical way of ordering words in English, typically involves saying the more agentive of the two nouns first. In passive sentences, the order of the nouns chicken and nurse is reversed. So you wind up saying the less salient or agentive thing first in most of our stimuli. And this non-canonical ordering of words has previously been shown to involve prefrontal regions like IFG and MFG, inferior and middle frontal gyri. And these regions form part of the multiple demand network, a domain general system that's active for a whole bunch of behaviors requiring general working memory and attention resources. And while there's a lot of hypotheses about the role of these regions, specifically in passive sentences, what exactly they're doing here remains a mystery. And to foreshadow, I think we'll shed some light on this. Okay, so let's look at results for passives. Here's an example sentence. Frankenstein is being hit by Dracula, and I'll show you the results from middle frontal gyrus, and which is one of these prefrontal regions that's disproportionately active during passives. So this time I'm gonna work backwards from the last word. So as the patient is saying Dracula, this classifier predicted Dracula, which we expect. During the production of the penultimate word, by, we are already detecting this upcoming word, which is neat because at the time that this is significant, you're not quite saying Dracula yet. Going back one more word, we don't see much. But then if we go back one more word to being, we're again detecting the object. And again during is, the second word. And in fact, before the patient even says the very first word of the sentence, prefrontal cortex is actively representing Dracula, the last word of the sentence, which the patient won't say until 6 words later. So what we're seeing in MFG is a fairly sustained representation of object specifically throughout the entire sentence. Now, this contrasts not only with what we saw for active sentences, but also with the same passive sentences, but in different brain regions. So here I'm showing you, I'm gonna show you the same sentences, but using classifiers trained in sensory motor cortex rather than middle frontal gyrus. And we might not be surprised to find that in sensory motor cortex, which is responsible for motor control and sensory feedback, we get the same kind of temporally congruent patterns that we saw for active sentences, where the blue line, the subject, Frankenstein, is significant at the beginning of the sentence when it's being said, and the green line is significant at the end. Okay, so now let's look at the aggregate pattern to take into account all ROIs. Here I'm showing you all significant results leading up to and just after the onset of the subject, and immediately we can see a difference. Whereas in active sentences, decoding was largely congruent. That is, we decoded the first word when it was being said and the second word when it was being said. In passives, we're getting a lot of both of these nouns, which you can see both in the smattering of significant green and blue lines throughout and in the density plot at the bottom. And during the production of the second noun, Dracula, we see a similar pattern, sustained encoding of both words throughout this entire period. Okay, so to get a better sense of the differences between actives and passives, we counted up the number of detections of the subject and object in each. So here I'm showing you just the number of classifiers where we significantly detected the subject while it was being said, what we see is that for both actives and passives, we get lots of hits. Now, if we look at the object, we see the same thing. For both actives and passives, we're detecting the object when it is being said. But here I'm just showing you congruent detections, the cases where we detected the word as it's being said. So next let's look at cases where we detect one word while the other is being said, incongruent detections. While patients were saying the object, in actives we detected 0 instances of the subject, but in passives we detected the subject in 12 classifiers, which out of 1,400 chances for detection is a highly significant increase. And similarly, while patients were producing subjects, we detected the object many more times during passives than actives. And remember, this last bar reflects detections of the very last word of the sentence while you're saying the very first word. So what's driving this difference between actives and passives? So far I haven't talked much about the spatial distribution of the information we're decoding, and that's because there hasn't really been much to say. Every one of our 7 ROIs, or regions of interest, contain information about words and with similar temporal profiles. But if we ask where in the brain we're detecting these incongruent nouns, we see a clear pattern emerge. The classifiers that detected incongruent objects, so again, objects decoded while the patient is saying the subject, these were primarily located in prefrontal cortex in IFG and MFG, the indigo and purple wedges. And in particular, the region with the most information about incongruent objects was middle frontal gyrus, MFG. In contrast, the classifiers that detected incongruent subjects came primarily from IFG, inferior frontal gyrus. And this is an especially cool finding for 2 reasons. First, it tells us what prefrontal cortex is up to during passives. If you'll recall, I mentioned there's this mystery about why prefrontal regions are highly active in non-canonical structures like passives. What are they doing? Well, here we have at least one answer. They're sustaining representations of words, specifically one word while you're saying another word. And the second cool thing is that we've unexpectedly stumbled on a potential answer to a central question in the neuroscience of language, which is how does the brain represent syntactic positions like subject and object? I'm not going to go into too much detail here because I think this needs more, we need to do more work here before we can make super strong claims, but suffice it to say there's a whole bunch of complex theories about how the brain might represent this stuff, ranging from things like excuse me, neural synchrony to interactions between power and different frequency bands. But here, we're seeing a really simple spatial code where subjects and objects are differentiated by which region they're represented in. And you'll recall that I foreshadowed this about a bit, pointing out that one of the remaining unanswered questions about sentence production is how syntactic roles are encoded. Now, I suspect that what we're seeing here is more likely maintenance of these words in working memory due to the difficulty of maybe producing passive sentences in this experiment, rather than anything purely syntactic. But still, the existence of a spatial code for something so complex I think is really intriguing, and we're currently doing some follow-up work to figure out how reliable this pattern is. Okay, so returning to our original question, are the neural dynamics of word production the same when words are produced in isolation as in picture naming and in sentences? And the answer is yes and no. Yes, because the neural representations of words generalized from picture naming to sentences, which we demonstrated by using classifiers trained only on picture naming data to successfully decode words during sentences. But no, because we also found a major difference in the temporal profiles of actives and passives. So unlike in actives, we decoded both nouns in passive sentences throughout the duration of the sentence. And this revealed a division of labor within the language system, where sensory and motor areas encode words in the order that they're being said, but in prefrontal regions like IFG and MFG, the order that words were processed in depended on the syntactic structure of the sentence. And then finally, we've got some insight into what prefrontal cortex is up to during passive sentences, as well as a potential clue as to how the brain might encode syntactic roles like subject and object. And with that, thank you all for listening. A big thank you to my PI, Adeen, and my lab. This project will be out in communications psychology relatively soon. In the meantime, there's a bioarchive that people can access. And I hope this isn't inappropriate, but a quick plug. I'm going through a bit of a funding crisis due to some of the chaos at the NIH. So if you're interested in this type of work and happen to have funding for a short-term postdoc, or if you're hiring assistant professors, I would be very interested in hearing about it. Okay, feel free to grab me a coffee. Thank you very much, I appreciate it.
Hi, thanks for the talk. Jack Dempsey, University of Illinois in Cascade Reading. I'm really interested in the implications of the passive sentences, obviously, how you get kind of this generation of both words. It says a lot about how we still care about the agent, no matter how it actually appears in surface form. And I was wondering whether you've done this or not, or what your thoughts are if you did do this, but biphrases and passives aren't necessary. So if you saw Dracula was hit by Frankenstein, you don't actually have to say by Frankenstein. And so I'm wondering what you would think, how much of this is the concept of the action taking place, and how much of it is actually planning, I have to produce this word in the biphrase?
Hey, it's a really cool question. Yeah, I would love to do that. We didn't, all of our sentences had biphrases. I'm actually thinking of doing this in Brazilian Portuguese with, they have two different kinds of passives, and one doesn't typically get the agent or the biphrase or whatever it is. And anyway, I'll keep you posted.
Hi, John Truswell, University of Pennsylvania. Hi. Terrific work. Wow is a good response to it. I think it's really excellent. I had a few questions and a suggestion. I guess the suggestion is one that just makes your life more miserable, which is you really should eye track these people while they're recording this stuff. Or you really should do decoding when they're not looking at the image versus when they are looking at the image. Because during the picture naming part, it's completely confounded because they're looking at Frankenstein while saying Frankenstein. And so the decoding may be in certain regions, or maybe even all the regions, actually a decoding of what they're looking at versus not what they're saying. And so it would be really cool to decode what they're looking at versus what they're saying, maybe in picture naming. Like you could get cases where there's nothing on the screen and they're saying Frankenstein. And use both of those decoders during the production phase, sentence production phase.
We did not do eye tracking. The thought of trying to implement eye tracking with ECOG is...
I think it would be easier to decode, do the decoding at the beginning.
But we did do two things that I didn't mention. One is there were multiple blocks of pretty much all of these tasks, and in one of them, for both sentence production and picture naming, the pictures were only on the screen for, we manipulated whether it was 200 or 300 milliseconds. Okay. And then in another block for picture naming specifically, we had the same characters or the same words, but just random images of chickens I found on the internet. So yes, I haven't actually looked at this yet because there's just mining this data set is going to take decades. But yeah, we can do that, and I really
encourage it. Yeah, it would be great. Thanks.
Hi. Hi. Sarah, Georgetown. I want to ask two questions, one's short, one maybe longer. The first question is really about your population. So these are epileptic patients, which means that they could have focal or global type manifestations of epilepsy, which could make me wonder how generalizable this would be in case that they are compensating using some other mechanism, a mechanism that you might not be mapping onto. So could you speak a little bit about your patients? And then the second is whether or not this is necessarily maintaining syntactic structural representations or maintaining conceptual representations. So it could be the case that they're maintaining these very dynamic representations, and it could be further complicated by the kinds of conceptual specificity of Dracula and Frankenstein compared to chicken and nurse. And so maybe one way of teasing apart is looking and seeing if you get modulations of the decoding based off of the conceptual specificity of the actors involved. Does that make sense?
Yeah, unfortunately, I mean, like I said in the talk, these stimuli weren't chosen with this analysis in mind. It would be really nice to be able to, like, isolate particular types of lexical representations, but there's just a ton of compounds. Like Dracula and Frankenstein, you might think is like a natural class semantically, but also they're 3 syllables with initial stress. So like, if you find, anyway. As for the population, it's all focal epilepsy because the patients are actually in there to identify the focus of the seizure so that you can then go back and take that part of the brain out. But yes, there is the possibility, and it's been shown that epilepsy does reorganize cortical networks. So, I mean, this is a problem with, I think, pretty much all ECOG work. You just don't know how much that's affecting things.
Yeah, I guess my question is whether or not the mapping site that you're doing is necessarily also the area of interest for their focal seizures.
I mean, so we don't choose where the electrodes go. That's chosen by the neurosurgeon and the neurologist. So yes, it's overlapping, but I'd say the majority of electrodes in these patients are not in ictal regions that are impacted by the seizures.
Awesome, thanks.
Yeah.
Thanks, Adam. John Brennan, University of Michigan. I guess want to ask about the spatial specificity of your analyses and whether there's an opportunity to look at more at a granular level at what the electrodes are doing. In part, and this relates to your kind of interesting findings about subjects and objects, actives and passives. So in the comprehension literature, decoding work with fMRI points to temporal lobe as having actually a pretty fine-grained spatial code for not active and not subject and object, but like agent and patient, these other thematic roles. But you're pointing at this sort of a rather gross or coarse-grained front. So the takeaway is first comprehension production and the existing literature on argument structure in temporal lobe, and second is whether this mismatch might be because you are looking at these very kind of large scale, these fine-grained patterns in large scale regions versus more fine-grained spatial code within a single region that you are collapsing over, and your thoughts on looking at individual electrode patterns. Yeah, thanks.
Okay. Wow, I have a ton of thoughts on this. I'll try and make this fast. We did not, unfortunately, I think all but one of our verbs were like typical agent patients. So unfortunately, it's gonna be really hard to disentangle with this data set anyway, what semantic and syntactic roles. I agree, regions of interest are just not the way to do this. It is, however, like a almost necessary simplifying, what's the word, way of doing this that you have for publication. Like, so initially, I did this in this incredibly complex but beautiful way where we started by creating dissimilarity matrices for each electrode so you get different sensitivities to the different sets of words. So for instance, in this middle matrix here, it's really tuned to the 2 middle characters. I don't know if you can see them, it's Dracula and Frankenstein. And again, this could be like a semantic electrode or it could be a like something that cares about having 3 syllables, I don't know. But yeah, so then instead of doing regions of interest, I clustered all of these representational dissimilarity matrices for all of the electrodes, and you get these really cool, using non-negative matrix factorization, doesn't matter. Anyway, you get these 9 clusters that are really cool. 6 of them really are just like a bunch of electrodes that care only about one word. So for instance, that top middle brain is all electrodes that only care about chicken and nothing else. And then the left 3 are these more complex patterns where you get electrodes that are different patterns for each word, electrodes that, we got a bunch that were sensitive to Dracula and Frankenstein, and then the bottom one is, what is that? Dracula and chicken. I don't know why those 2 go together naturally. But anyway, yeah, so I think this is a better way of doing this. I don't think I could ever get this published in a neuroscience journal because they just don't care. I have tried, yeah, I know, it's really unfortunate. So maybe, I don't know. Thank you.
Thank you for your talk. I think, continue on this similar thread as previous talks, previous questions. I'm actually more surprised that you don't get object decoding in the active sentences until like getting really close to the production of the object itself. And so I was wondering what that says about, I guess, if you consider maybe the behavioral literature on active versus passive sentence planning, what that tells you about maybe the types of representations that are being decoded for the objects in your experiment.
So you're pointing out that, let me see if I can find it here. No. Yeah, is this?
like in the active sentences where like, there's not as big of
Oh.
a smattering, like,
Oh, okay. Yeah. I don't know where that is. Here. So you're saying there's fewer subjects that were decoded?
yeah, or I guess I'm surprised that like Frankenstein isn't that you're not able to decode it earlier on in like
Oh.
the sentence.
Yeah, that's also interesting. I don't know. I did try time locking this to the onset of the stimulus, thinking, okay, maybe in the very beginning you're processing both, and I honestly don't know why we're not getting it. I had the same thought. It might be a power issue. There were only, for some of these patients, like 60 sentences that we collected, because just constraints with working with patient populations, but yeah.
Thank
Good
you.
question, though, yeah.
Laurel Brum, UC Santa Barbara. This is awesome.
Thank you.
You started with the two problems of doing sentence production neuroscience stuff. Turns out it's easier to look at someone's brain and get spatiotemporally locked things than to get them to say what you want them to say, which I'm not surprised by. But can you leverage, so you had this very cool analysis where you looked at, predict this thing, compare that thing, look at when it gets it wrong. What about, using a classifier of the sentences that people, like the kind of canonical sentence types, the active passive, and then seeing what happens with the weird productions? Have you done that? Would that give you any interesting data?
Wait, sorry, weird productions how?
So when people say the hard to code productions, like when they omit arguments, or when they give you a long story, or when they give you an impoverished whatever, yeah.
This task, okay, so the trade-off in production is how
Yeah.
ecologically valid it is and how good the data are, right? And so this was very much not ecologically valid. We pretty heavily constrained things, and we didn't really get much variation in what people said, aside from the fact that there were like 2 to 3 times more active sentences than passive sentences. We didn't enforce, yeah.
Okay, cool.
Yeah, sorry, that would be really cool to look
That
at.
would be really cool, right?
Yeah.